Well, hello everyone. This is Dawn Van Dam, and I'm the General Manager of Cambridge Health Tech Associates. Welcome to today's webinar on Back to Basics, an overview of RNA-Seq. So we have two terrific speakers today, and I'm just going to give a little background information on each of their talks. If I could just ask everyone who's online to mute their phone, that would be great, so we don't have as much background noise. So Dr. Colin Dewey will be speaking about one revolutionary aspect of RNA-Seq technology and the fact that it is its ability to provide information regarding transcript abundances in species whose genomes have not yet been sequenced. In such situations, transcript quantification involves a combination of de novo assembly and abundance estimation, both of which are challenging tasks when a reference genome is unavailable. Dr. Dewey will describe the basic steps. Our other speaker, Dr. Yulong Louis, will give a brief introduction of the bioinformatics considerations of deriving allele-specific expression from RNA-Seq data. He will further explain his group's efforts in integrating transcription factor binding prediction to identify functional regulatory variants through allele-specific expression data. So we're really looking forward to both of our speakers today. Just to give you a little bit of background on Cambridge Health Tech Associates, although we are providing this webinar today for free, we also do provide services to help transform science into commercial success. We provide services in consulting and market research, in consortia and collaborations, particularly around doing technology evaluations, and then also in marketing and communication, helping people be successful in bringing new products to the market.
I chose this proposal over this.
Another area that we specialize in is we manage a technology evaluation consortium where we have between 10 to 15 major biopharmaceutical companies, and they help look at and evaluate and give feedback on between 4 to 8 new technologies every year. So we are actually looking for new technologies to evaluate in 2014, and also to have more pharmaceutical companies join this technology evaluation consortium. So if you'd like more information about it, we encourage you to let us know. That would be fabulous. So let me just give you a brief introduction today about each of our speakers. Dr. Colin Dewey is Associate Professor of Biostatistics and Medical Informatics at the University of Wisconsin-Madison. Before joining the faculty at UW-Madison in 2006, he earned both his Bachelor of Science and PhD from the University of California, Berkeley in Electrical Engineering and Computer Sciences with a designated emphasis in computational and genomic biology. Colin's lab develops computational and statistical methods for genomics with an emphasis on sequence analysis problems. A major focus of his current work is the development of methods for analyzing RNA-Seq data, particularly those generated from organisms with a sequence genome. In addition, he is interested in the problem of large-scale sequence alignment and specifically methods for aligning whole genomes. Supporting his research interests, Colin has participated in the ENCODE Consortium and the International Sequencing Project for the mouse, rat, and chicken genomes. Dr. Yulong Lee is an associate professor in medical and molecular genetics at Indiana University School of Medicine. Also a member in the Center for Computational Biology and Bioinformatics and Center for Medical Genomics. Dr. Louis' research area is to use a systems biology approach to study gene regulation, including transcriptional regulation, post-transcriptional regulation, and epigenetic regulation. Dr. Liu's group is specialized in data processing, analysis, and integration of next-generation sequencing data from multi-omics domains. So without any further ado, I'm going to turn the presentation over to our speakers today. And also remind you that if you have any questions, if you could, for the speakers, if you could put them into the chat box, and then I will ask the questions of the speakers at the end of their presentation. So we're going to turn it over now to Dr. Colin Dewey to talk about his presentation today. Thank you.
Great, thank you very much, Dawn, and thank you for having me to give this talk. So today I'll give you a little bit of a basic overview of RNA-Seq analysis and non-model organisms, and I'll tell you a little bit about sort of the application that is driving our particular group's work. But the problems that we're tackling with this are sort of very general and can be applied in many situations, including in the model organism case when you do have a genome sequence. Okay, 1 second while I figure out my controls here. Okay.
Sorry, Colin, I just passed the ball to you. And also, could you just repeat what you said? Because I think some people were unable to hear a minute ago. So if you could just repeat your initial introduction, that would be great.
Okay, I'm sorry. Yeah, so thanks again for having me to speak. And I'll be talking today about the basics of doing RNA-Seq analysis in species that do not have a sequenced genome. And I'll tell you a little bit about the application that is driving my group's work for this, and the methods that we're developing in this area are applicable to as many organisms which share this situation. But also, I would emphasize that the things that we're developing also are applicable to the model organism-based situation. So I'll begin just by talking about the example application that's motivating my group, but it's also going to maybe serve as a motivating example for all of you right now, which is the study of the salamander axolotl, whose scientific name is Ambystoma mexicanum, and as its scientific name suggests, it's originally from Mexico, and its natural habitats were in a number of lakes throughout Mexico that are now, unfortunately, severely hindered and are such that the axolotl is essentially an endangered species at this point. And in fact, it's more commonly found now in the fish tanks of people throughout the world, so it's sort of commonly sold as a pet. It has a number of interesting features, one of them being that it's a neotenous amphibian that retains its juvenile form throughout most of its life, so it stays in the water pretty much all the time. But the interesting part for us and for our collaborators in James Thompson's lab here at UW-Madison are the salamander's amazing regenerative abilities. So this salamander has the ability to regenerate parts of its limbs, heart, brain, and even its tail and spinal cord if those become injured or amputated. Okay, so there are a number of huge challenges with analyzing sort of the molecular biology of such species, and in particular what we're interested in is basically what are the genes that are responsible for the amazing regenerative ability of the axolotl salamander. So how do we get at that? Well, one thing that's making this very difficult is that the genome of the axolotl has not been sequenced, and as are the genomes of many non-model organisms. And the reason for that for an axolotl's case is that its genome is about 10 times larger than that of the human genome, so that makes it a little bit difficult for assembly. In addition, often you have very incomplete or even no gene annotations whatsoever for such species, and for what RNA information you do have, it's usually so limited that you don't really have enough to really make a comprehensive microarray or a comprehensive set of PCR primers that can be used to do quantitative PCR, say, of genes of interest. So that's where RNA-Seq technology really comes to the rescue, and that is really revolutionizing this particular type of application. So I'll give a little bit of an overview of RNA-Seq technology just in general, but for all cases. Typically one starts out with a sample pool of RNA shown on the left. And there are a number of different protocols that are used for RNA-Seq, but the one that I'll show is kind of maybe the more standard one right now. And in that one, you start out by first fragmenting your RNA into a number of small RNA fragments. Then through both reverse transcription and an amplification step, you obtain a set of amplified short cDNA fragments that are derived from that RNA. Here, so the slide is a little bit garbled in the transition from Mac to PC, but the cDNA fragments are then fed into the sequencing machine, which typically only can handle DNA, and they output a set of short reads. They're typically in the order of 35 to 150 base pairs long that are derived from the ends of these cDNA fragments.
Okay. Can you send me these numbers?
So there are a number of other protocol steps that can be added here that, depending on the application, one might be to do poly-A selection near the beginning, which will restrict your analysis to only those transcripts that are polyadenylated, which typically the protein coding fraction of the transcriptome. And there might also be a size selection step near the end, which restricts to those size RNA fragments that you're interested in. Typically, those are restricted to maybe 200 to 300 base pairs in length. Just some basics for Illumina fragment sequencing. There's some terminology here that's important. So I told you before that what we're actually sequencing are the ends of these cDNA fragments. Each fragment is typically 200 to 300 base pairs long. And what the sequencer is doing is actually just reading off one end, in the single end case, of this fragment. And that fragment, that piece that we read off is called the read, and that's usually much shorter than the fragment itself. We also have data called paired data. Yes?
Colin, sorry to interrupt you, but there's quite a bit of background noise. I'm just going to ask people if they can mute their phones because there's something happening to the system. So if just put their hands over their phone or put on mute, that would be great. They were trying to do a master mute as well, but for some reason it's not working well today. So, okay, go ahead, proceed, Colin. Thank you.
Okay. So I hope you all have heard some of what I've said. So I'm going over right now the Illumina fragment sequencing sort of jargon about what type of data gets generated. So I talked about single end data, which come from, or reads that come from one end of each fragment, and paired end data are simply those data where you actually get a read off of both ends of the same fragment, and those two reads are linked together in the output, so you know that they were derived from that same fragment. And this provides you with extra information that can be used to help you know, an assistant in telling you where exactly that fragment came from within the transcriptome, or help you with actually assembling the RNAs. Okay, I'm sorry. So this slide is also a little bit garbled. This was supposed to show the sort of actually a piece of an actual data file. It's a FASTQ file that contains a set of reads from a single RNA-Seq experiment. And in these files, each read is represented by a number of lines that give you the sequence. There's a name identifier, and there's also a quality score string, which is basically the sequencer's way of telling you its confidence in the base calls that it made at each position within each read. And each lane of, say, Illumina technology might give you 20 to 200 million reads in these files. Okay. So now I'll get to the computational side of RNA-Seq, specifically for studying the transcriptomes of non-model organisms. So here the basic idea is that we're first going to feed our reads into what we refer to as usually a de novo transcriptome assembler, which is given the task of actually reconstructing the full-length RNAs from that set of reads. We then feed those transcript sequences and reads into a transcript quantification method, which will actually quantify the abundances of each of those transcripts. So if we're interested in actually what genes are going up or down, this is ultimately what we need. And lastly, at the end of the day, often what we're interested in is changes in gene expression between different tissues or across time or across different treatments. And for this, we use differential expression methods that take in as input the transcript abundances from a number of samples, and then we'll output a subset of those transcripts that look to be differentially expressed across those inputs. So a number of challenges with this protocol, especially in the de novo case, and those are the two challenges that I'll discuss today. So one is how do you actually do accurate quantification with these sort of data sets? And the second is maybe kind of a more philosophical one is are transcript sequences really the things that we desire as the us what we're assembling and quantifying, or does those make sense as what we need to be working with here? Okay, so first I'll start with the quantification issue. So in here's sort of the basic flow of quantification with RNA-Seq. So typically you align your set of reads to the transcript set that you have, so either from a de novo assembler, or if you're in the model organism case and you have a genome, you usually have an annotated reference there. Then typically you're going to keep only those alignments that have a few mismatches or indels in them, so you only want to look at sort of high quality alignments of each read. These are then sort of essentially just counted. The most simplest thing to do is to count the number of reads that align to each transcript, and then you sort of use those counts to essentially come up with relative abundances of each of those transcripts. And to do that, you have to normalize these counts in a number of ways. One which is to normalize by the length of each transcript, because the RNA-Seq protocol, just by its nature, tends to produce more fragments for longer transcripts because of the way that the fragmentation and priming occurs. And then you also need to normalize by sort of all of the transcripts at once to get at essentially transcript fractions for each transcript. So each transcript occupies some percentage of the full transcriptome, and that's ultimately what you're estimating with RNA-Seq. So that looks fairly straightforward. But there's one step here that I want to emphasize, and that's sort of often glossed over, which is that people often will remove all of the reads that have more than one good alignment in this step. So if a read maps to multiple transcripts equally well, then those will just be thrown away because they tend to complicate the analyses. Okay, so is this really a problem? Maybe it's not a big deal to throw these reads away, but actually it is kind of a big deal depending on your application. So if you look at, say, in the mouse, if you had read lengths of length 25, you'd have maybe 17% of your reads that are mapping non-uniquely. If you increase the read length to 75, you might drop that down to 10%. And if you go higher, you can get that even lower, but it doesn't go to 0. And the reason for that is that there are a lot of sort of duplicated genes in the genome and or repetitive regions in the genome. In other species, such as maize, corn, if you have read length 25, you get more than half of your reads being multi-mapping reads, and this sort of is testament to the repetitive nature of that genome. In our case, in the axolotl genome with read lengths of 76, we get almost a quarter of our reads that are multi-mapping. Okay, so what's the problem here? So we don't really want to throw these things away, because doing so would cause us to lose information about abundances, and also potentially bias abundances if we don't do the quantification carefully. So to address this issue, we derived a method back in 2010 called RSEM, which is essentially a statistical model-based approach to doing transcript abundance estimation from RNA-Seq data that takes into account reads that map to multiple locations, both between multiple alternative spliceomes of the same gene and also between different genes in the genome that happen to share a lot of their sequence. So I don't have time to go through all the details of the model, but it's essentially a model that takes into account a number of different features of RNA-Seq data, including the lengths of the fragments, the true transcript from which each read is derived, the start positions of those reads, quality scores, et cetera. And at the end of the day, what it's doing is it's finding maximum likelihood estimates of abundances of each transcript, and to do so, it uses a commonly used algorithm called the expectation maximization algorithm. Okay, so I'll briefly just show some advantages of using such a method. So we also compared to a number of other methods that were out at roughly the same time that was published. And in these plots, what we've done is, this is from simulated data where we know the true abundances of each transcript. And here the x-axis here is a sort of a level of error in your transcript abundance estimates. And the y-axis is the percent of genes, or on the right, isoforms that whose estimates were greater than that threshold. So ideally, you want to have these curves near the bottom left-hand corner. And we see here that RSEM tends to get closest to that corner in predicting fewer numbers of genes and isoforms that have error much higher than certain levels. Okay, another thing that we've done with RSEM is to really look at how do you design RNA-Seq experiments to get the most accurate quantifications. And here we considered the scenario in which you had a fixed budget for the number of bases that you were able to sequence in a very simple economic model, which you pay per base. And we asked what's the right combination of read number versus read length that would get you the best accuracy. Okay, so we considered 4 different experimental settings. One which would just be a standard 35-base single-end read experiment in blue, and then 3 other experiments that would have twice as much sequencing power in terms of number of bases. One that would be 70-base single-end reads, one that would be 35-base paired-end reads, and the last would just be simply double the number of 35-base single-end reads. So these last 3 experiments all have the same throughput and twice as much as the first. Okay, and we looked at both the gene isoform and isoform levels sort of relatively within each gene and looked at the percent error of the estimates that you would get in those cases. And as you expect, so the ones that have double the throughput have lower errors. But surprisingly, the one that does the best, at least at the gene and isoform levels, is just simply the double the number of reads for short reads, for 35-base single-end reads. And that's sort of just testament to that it's really the number of reads that is sort of more important for quantification than the read length itself. The story is a little bit more complicated when you're looking specifically at individual genes and looking at the relative abundances of individual isoforms within those genes. Their paired-end data does look to be a little bit better at distinguishing between those isoforms, and that sort of makes sense because these longer paired-end reads tend to tell you sort of combinations of exons or splice functions that are occurring in your transcripts. Okay, so the last topic I'll talk about is looking at what can we actually assemble from RNA-Seq data, and are we really trying to look at individual transcripts? So if anyone who's familiar with these assemblers knows that they underneath are using these sort of large graph structures. Common ones used are the De Bruijn graphs. And transcript sequences that are output are typically paths to these graphs. Okay, so there's a number of challenges with this whole approach, and one is that there are often a huge number of paths to this graph, and this is made complicated by genes such as Drosophila DSCAM that has more than 38,000 different isoforms for the same gene. So that's just an enormous number of paths to that graph. And the second is maybe a statistical issue, which essentially says that even if you knew all the different isoforms of your genes, you may not actually be able to estimate the abundances of those isoforms with any certainty because there's a lot of because of the combinatorics of how these isoforms share their sequence. Okay, so in this simple example I have at the bottom, it actually turns out that for short RNA-Seq data, you cannot actually estimate the abundances of these 4 isoforms. Okay, so the solution that we've come up with recently for this was to actually just use the graphs that are generated here. And we're using a type of model called a probabilistic splice graph, which are based on splice graph structures that have been used for more than a decade now to represent alternative splicing in genes. And here the idea is that what we'll do is essentially we'll just use the graph for each gene and now annotate the edges of that graph with probabilities that represent conditional probabilities of transitioning from one exon to another and to complete a full transcript. So here what happens is that in this example here, with just 2 sets of probabilities, if I take the product of these probabilities along each path, I get a relative frequency for that isoform within your sample. Okay, and splice graphs can have many different forms that can take into advantage different levels of dependencies between different splicing events. And it's a sort of open research question how to go from the raw data to the appropriate splice graph that represents the proper dependencies between exons and splicing events. So what we've done is to contribute to this sort of generative model to the literature now for RNA-Seq analysis. And we have an efficient method that takes advantage of dynamic programming to actually compute these estimates on these graphs efficiently, even when there are an exponential number of possible isoforms per gene. Okay, so I just want to briefly jump back to the axolotl just to kind of show you some interesting results we're obtaining from applying these methods. So what we've done is the following experiments where we've essentially amputated the forelimb of an axolotl and allowed it to regenerate over a course of several months. And what we've done now is to then take RNA samples of the regenerating limb at a number of time points, actually over a dozen time points, and to look at what genes are sort of going up or down at each of these different time steps. And the one result that I'd like to highlight that is sort of exciting is, and that sort of supports older literature, is that early in this time course, so here I have a plot of the time points on the x-axis, and the y-axis being a percent of upregulated genes, is that a big fraction of the upregulated genes near the beginning of this time course belong to the class of genes that we refer to as oncogenes, or the genes that are sort of the ones that are often causative of cancer. Okay, so there seems to be a large burst of these oncogenes near the early, at the early time points of regeneration, and then it slowly tails off over the course of about a month until it's back to its sort of baseline level. And this is supported by older literature that has sort of seen a bit of a link between regeneration and cancer. The picture on this slide is depicting an experiment in which a mutagen was applied to the skin of another species of salamander, and instead of causing a tumor growth, this salamander actually just grows another limb out of that part of the mutagenized limb. Okay, so just to summarize, RNA-Seq is really just an incredible technology for studying these model organisms, especially ones that have these really cool abilities, including regeneration. And our group is doing a number of solutions to address some challenges in the analysis of RNA-Seq in these situations, including accurate quantification when you have these reads that don't map uniquely anywhere, and then also some machinery for actually doing quantification when you have very large sets of transcripts that maybe are best represented as splicing graphs. And lastly, we're now using these to study this regeneration process in axolotl and seeing this sort of interesting connection between cancer and regeneration. So with that, I'll leave you with a set of references that you can refer to if you want to learn more about the methods that we've developed and the results that we've obtained thus far in the axolotl salamander.
Great. Well, thank you so much, Colin. That was terrific. And again, we apologize for the early on difficulties in hearing the audio, but there will be a recording of this that we will send out to everyone afterwards. So, Colin, if you could just switch the ball to Yunlong, we will move it over to his presentation, and I will just remind everyone in the meantime that they can put questions into the chat box, and then I will ask the questions once the two presentations are finished. So over to you, Yunlong.
And thank you very much for Dr. Dewey for the wonderful introduction of the RNA sequencing experiment. So I'm going to switch the gear a little bit, talking about allele-specific expression studies from RNA sequencing data. And I will first briefly introduce what is alternative allele-specific expression and its role in studying gene regulation. And I will go ahead to talk about opportunities of challenges using RNA sequencing technology, and mention a little bit about the power identification, and also towards the end introduce briefly about integrating transcription factor binding analysis to predict the functional cis-acting regulatory variants. So first part, what is allele-specific expression? Allele-specific expression is the phenomenon that transcripts from maternal and paternal alleles express at different levels during transcription. In this particular hypothetical example, you can see clearly that there are two alleles here, and the one in the coding region, there is a heterozygous variant, which is A allele or C allele. In this example, you can see clearly that A allele expresses more than the C allele, and this is the allele-specific expression. Usually, there are multiple mechanisms that can cause two different alleles expressed differently, but most notably will be the genetic variants in the regulatory region, including the enhancer regions, promoter regions, or 3' UTR regions, or sometimes allele-specific epigenetic patterns. So this is the allele-specific expression. So why we study allele-specific expression? And we all know that gene expression is a very complex trait that is influenced by a combination of genetic and environmental factors. Even for the genetic factors, that will include the cis-acting and the trans-acting genetic and epigenetic variations. The nice thing about allele-specific expression analysis is that it can eliminate environmental and transacting effects because the two alleles serves as a within sample control to each other. For example, when we know that when we study a human, the data from human subjects, we often observe a very big variability. And this is because our living environment is different. There we have different environmental exposures, and people use different drugs and different exercise habits. And so that will cause a big variability in the gene expression levels. And it is very common that we often see we need dozens or sometimes even hundreds of samples to measure the gene expression levels from one particular tissue. However, when we look at allele-specific expression, these two alleles, they live in the same cell, so which means they have been exposed to the same environmental effects, and also to a larger extent, they also exposed to the same transacting factors, which means that all the expression differences between these two alleles has to come from the cis-acting regulation, which is the genetic variance in the regulatory region or epigenetic effects that is associated to allele-specific effects. Therefore, allele-specific expression is a very powerful tool to study the gene regulation. This is just a hypothetical example explaining why RNA sequencing experiment can be used to study allele-specific expression. So from this pattern, we can see this particular location, and the middle part is a G, and actually it's a heterozygous SNP. It can be either G or A. And from the RNA sequencing data, and we can see clearly there's a 7G there and 3A here. And without thinking too much about statistics, we might have the impression that allele frequency for the alternative allele, which is A allele in this scenario, will be about 30%, and the allele imbalance is 7/3. So this is just this is not put into any statistical framework, but just to give you the impression why RNA sequencing data can be used to study allele-specific expression. And there's a couple of challenges to do this. The first challenge is the data availability. It is not always true, but preferably requires both DNA and RNA sequencing data from the same individual. So because if we want to identify allele-specific difference, we'll first focus on the heterozygous variants, and the easiest way to identify heterozygous variants is from the DNA sequencing data. One may ask that since the RNA sequencing transcripts already have the variants here, can I just go ahead to call the heterozygous variants from the RNA sequencing data? For example, the example listed here, you can see clearly this is a heterozygous variant that has A and G alleles. And the answer for that is yes, you can, but sometimes in some situations, this may cause some of the problem. For example, if A allele, if this in the DNA level, this location is a heterozygous variant with A and G alleles, but A allele did not express at all. So what you observe is only the G allele. From the RNA sequencing alone, you may make the wrong conclusion that this is a homozygous reference. There's no heterozygous variance there, so which limits the power of studying allele-specific expression. And the second challenge actually is on the bioinformatics part, because most of the popular alignment algorithms are biased towards the reference alleles. And so I will get into this a little bit more and to explain why this happens. And the third one is the statistics. And so actually, in order to identify a subtle difference in terms of expression difference between two alleles, the subtle means 1.5-fold difference, less than that. And we may need hundreds of sequencing rates per base to be able to call the allele-specific expression more accurately. So I will have one slide talk about the power calculation or sample size justification for that. So coming back to the second point, which is about informatics part, most popular alignment algorithms are biased towards the reference alleles. Okay, so there are a couple, so this is no big deal for the DNA sequencing analysis. When people try to identify the genetic variants, it's either the homozygous reference heterozygous alternative or heterozygous reference. So basically, your alternative allele is going to be 0%, 50%, or 100% of all the sequencing rates you observe. However, for RNA sequencing analysis, this is a very big deal because we are trying to quantify the differences between the expression level of two alleles. And that subtle advantage that is offered by the alignment algorithm can really generate a lot of values for our analysis. So in order to address this, people propose several different ways to using different level of bioinformatics analysis to correct the alignment-specific bias. The first one is to align the sequencing rates to both the parental genomes. And apparently this is only good for the animal studies. And we know that for many mouse projects, for example, mouse strains, the parental strains, their DNA sequences are available. So if we want to study the allele-specific expression of the F1 offsprings, we can really map the sequencing rates back to both two parental strains and further consolidate our results. And this will be a very powerful analysis that can almost completely remove the alignment-specific bias. However, this type of approach is not always possible, and especially for human studies. So the second general approach is to use your regular alignment algorithm and further do the post-mapping correction for the RNA sequencing data. Again, this is another hypothetical example. For this location, for the DNA part, you can see this is a heterozygous variant, and circle is the reference, and cross is the alternative. If we focus on this particular location, when we do the sequencing mapping, we allow, at this scenario, up to 2 mismatches for each individual sequencing rates. Okay. However, if we don't do any consideration on this, we just count the number of rates that represent the reference or alternative, we may have the conclusion that there are 6 rates support the reference and 4 support alternative. And you may have the slight hint that this can be allele-specific expression. The expression levels of 2 alleles can be different. However, the red rates need to be discarded because the alignment algorithm actually gave additional advantage to it because everybody else has only 1 mismatch beyond the variant loci. And so after this correction, that removing these red rates, we might have just 4 reference and 4 alternatives. So the bias is completely gone. So we have done this for several different studies, and we found that this analysis is reasonably powerful in terms of removing some of the algorithm-specific bias. And a third approach is to use an alignment algorithm that allows IUB code or designed for including alternative alleles at known polymorphic sizes. There are quite a few algorithms that provide that options, including the very popular BWA and a few other algorithms. So when we study allele-specific expression, you may think to turn on those options. And the second, another important issue is the power calculation. So in this case, for example, if F stands for the alternative allele frequency, and our null hypothesis actually is F is equal to 0.5, which means there is no imbalance across both 2 alleles. And there are several different ways to do the sample size calculation, including some of more frequencies way or Bayesian-based approaches. I don't have time to go through those, but this slide just show you one figure of our calculation. And from this slide, you can see clearly that we need 101 sequencing rates to be able to have 90% of power to identify the allele imbalance at 2, which means one allele is 66%, the other allele is 33% at the p-value at 5% level. And even we reduce the power to 80%, we still need about 75 sequencing rates to cover that particular location. And as people work on a lot on the RNA sequencing experiment, we know that for some of the very highly expressed genes, probably 100 sequencing reads to cover one genomic location is not a big deal. But this is not true for most of the genes, which it has a moderate or somehow low expressed levels. If we really want to capture the low expressed or moderate expressed genes, their allele-specific effects, we really need to think of increase the sequencing depth. So those are the considerations you might need to have at the experimental design phase. And another issue is when we try to study allele-specific expression, and we have to rely on the presence of heterozygous variants in the coding region. And if there's one gene in the coding region, there's no heterozygous variants, and we can pretty much forget about identifying allele-specific expression. But when we go back to the 1,000 Genomes Project, based on the DNA sequencing, identify the heterozygous variants location for one individual, and for most other individuals, we did this tally, and we found over 10,000 genes do not have any heterozygous variants in the exon region of one individual. So if our analysis is based on one individual RNA sequencing data, this 10,000 genes, regardless how much they express, and you are not going to identify the allele-specific expression. So these are the considerations you might need to have before you think of using RNA sequencing data to do the allele-specific expression studies. Okay. Now I'm going to use a couple of slides to talk about why we can learn, what we can learn from the allele-specific expression. Actually, the goal for that is to study gene regulation. As we can see from this hypothetical example again, and the top allele and bottom allele, in the coding region, there is one heterozygous variant, which is C or A. And clearly, the bottom allele expresses more than the top allele in this case. And the reason for that can vary. However, most profound reason could be there is a genetic variance in the regulatory region, in this case, the promoter region, and there is an A allele or G allele, and that will affect the transcription factor binding in an allele-specific fashion. And so the question is, can we use the allele-specific expression identifier from the RNA sequencing data and trying to further infer which heterozygous variants in the regulatory region that can potentially cause the allele-specific expression. And this will help us to identify the functional genetic variants in the regulatory regions. And in order to do this, there are two different approaches that we need to take. And they both need it. The first one is, in order to know that which allele in the promoter region or regulatory region and is in the same haplotype with the coding allele, we need to first face the regulatory and coding variants. We need to make sure, in this case, A in the promoter is corresponding to the C in the coding region. And otherwise, we may not be able to put the study in a very meaningful way. And so we know that from the RNA sequencing data, we can only measure the variants in the transcript region. So the heterozygous in the promoter region may not be measured, even for the DNA sequencing. And if for the two variants, they are pretty far away from each other, we may not be able to measure them and very effectively. So the phasing is required in order to proceed this type of study. And the second general approach we need to take is we need to do the transcription factor binding motif analysis, and in this case, to speculate whether A allele and G allele will affect the transcription factor binding. So this is the general schematics of how to study the functional variants from allele-specific expression. Let me give you a very brief example that we have studied, that this is a combination of the DNA sequencing and RNA sequencing data of 23 individuals. In these 23 individuals, we identified 730 variants showed significant imbalance between the alleles in at least one sample. So the blue part is the lower expression in the alternative allele, and the red is higher in the alternative allele. As you can see that although this has a lot of although this has so for individuals there's a lot of variabilities. However, there is general trend that this allele-specific expression can be replicated across multiple individuals. So when we turn back to the transcription factor studies, and this is one example, and we found that this is the CMAC binding protein, and which is transcription factor that is very important in a lot of physiological situations. And we found there are 50 heterozygous variants on this CMAC binding sites, and 28 of them showed significant allele imbalance. From this figure, you can see that X corresponding to the transcription factor changes by the promoter variants, where in the right part it represents the gain of the binding, and the left part is the loss of the binding. And the Y here is representing the allele imbalance actually caused by the identified by the RNA sequencing data. On the top part, it will be higher expression in the alternative allele, and bottom is a higher expression in the reference allele. From this figure, we can see clearly that there is a positive trend of those blue dots, which means that all these variants support that this CMAC is an activating factor that will increase the gene expression in this particular individual. And this is a reverse example that a situation of a negative regulatory transcription factor, which is NR2F1 binding sites, and we have seen that there are 17 variants that support NR2F1 is a negative transcription factor. So these are some of the examples that you can see clearly that we can using this deeper bioinformatics analysis to understand how the transcription factor works in this particular scenario. And the overall effect for this example will be we identified one negative transcription factor and actually three additional positive factors that will increase the gene expression levels. Okay, so the summary for this part is the promoter variants affect gene expression by changing the binding affinity of a transcription factor, and allele-specific expression identification from RNA sequencing data requires careful analysis for quantitative evaluation. And combining DNA sequencing data with RNA sequencing expression data will provide a powerful tool in studying functional regulatory variants. And that's the end of my talk.
Great. Well, thank you so very much. I want to say thank you both to Dr. Colin Dewey and also to Dr. Yunlong Lui. I'm going to open it up for questions, and I have a couple that have come through, so I will start them shortly. I just need the slide advanced, one slide. So maybe the first question I will refer to Colin. So Colin, what is the current state of de novo transcriptome assembly methods, and how should one choose between them?
Great, thanks for that question. Yeah, so currently there are a number of de novo transcriptome assembly methods out there, and although I would say that's still sort of the early stages of de novo transcriptome assembly, and there are sort of new advances coming out every day. So some popular ones are Trinity, Oases, Transipis, SOAP de novo trans. These are some of the ones that we've looked at. And I would say it's just currently a very difficult situation because you have a lot of different assemblers, and each of these assemblers has a huge number of different parameters that you can all vary. So you just have no clue when you're running your assembly, which assembler do I use and which set of parameters do I use? And you don't know if the assembler that was did well on, say, assembling the human transcriptome is best for your particular species that you're looking at. So one thing that I would just say is to look at how your reads, how your actual data are aligning back to your the transcripts that are assembled, and look to see that your transcriptome is explaining the actual data that you have. And to that end, our group is actually developing a method for evaluating some of these de novo assemblers. So be on the lookout for a method called RSEM-eval soon, which will allow you to actually evaluate a given assembly given the reads that you use to assemble it.
Okay, great. And would there be anything that you would like to add, Dr. Yunlong Lui?
So there's a question coming out that how do you get TF binding info for each variant? And I think that's a very good question, and I haven't had enough time to explain that in the slides. And the transcription factor binding is a very mature field currently. So what people usually do is rely on the position weight matrix or position specific matrix, scoring matrix from either Transfact or JASIC database. And so all these will be used to predict a short stretch of sequence whether there's a binding likelihood of this transcription factor. And some people will just look at whether this transcription factor is really overlapping with the variants. But a better approach will be to look at before and after the genetic variants, whether the transcription factor affinity score will change significantly. So there are several different approaches, published studies that is trying to address this situation, and that will be the ones that we can use to make the prediction.
Okay, well, while we have you speaking right now, why don't we ask you another question? What alignment algorithm is developed for allele-specific expression analysis?
Thanks for the question for this one. And actually, the current the status is most of the common alignment algorithms do have the option that you use the IUB code, or you use some of the variants that some of the algorithms, and when you do the mapping, and it can be sensitive to the alleles to the multi-polymorphic sites. So most notably, if we talk about BWA, they do have the parameter to mask those sites that we know they are heterozygous variants. And also the same thing will be in the solid data, which is the color space. And if we look at life scope mapping suite, and they do have the same option to do to mask the variant loci. However, we really need to be very careful about that part, because when you mask the variant location, if two variant locations they are very close to each other, or multiple variant locations they are very close to each other, even this approach will not work. And also if when we do the RNA sequencing alignment, if the DNA sequencing for the specific individual is not available, meaning that the variant is not specific to this particular individual, some people will just go ahead to mask all the variants in the 1,000 Genomes Project or in the DBSNP, and that ends up with too many variants locations will be masked out, and then that really reduces our capacity or ability to detect, to have the mapping done properly. So the short part of this answer will be most of the alignment algorithm can address the situation, but when we use that, we need to be very careful.
Okay, great. Thank you very much. I just want to remind everyone as well that there is a survey link that's been put into the chat box, and we would love you to go and answer that survey just so we can get some feedback on the presentation today, and also get your opinion on what you'd like to see for future webinars. And also to remind everyone that we'd love to have some more questions, but I will turn back to Colin for a minute and ask Colin the next question. So in your study, you performed some functional analysis on the genes that were upregulated during regeneration. How does one perform such analyses if the transcripts were generated by a de novo assembler and are unannotated?
Great, thanks for that question, Don. Yeah, so this is something I sort of glossed over during my talk, and that is that when you throw these reads into the transcriptome assembler, and then you get these transcripts, and then you want to do differential expression analyses, you might find that a bunch of transcripts are differentially expressed, but you have no idea what those transcripts are in order to do any sort of interesting like go term analyses or that sort of thing. So what we did in our study, and this is a common approach now, is to sort of map your transcript to some other reference organism, or use BLAST somehow to kind of annotate each of your transcripts with its sort of closest homologue with another species. And so in our case, we actually used the human genome as the reference sort of protein set, and so we essentially aligned each of our transcripts against the human protein set to annotate them with the current human annotation, and then performed all of our statistical analyses sort of assuming that we were sort of using the human genome as the reference. Of course, this has the major drawback that if there is anything specific to axolotl that, like, or specific genes that are not present in human or whatever reference species you're using, you're not going to see that. But unfortunately, that's sort of the drawback that you're going to face all the time if you haven't actually gone in and characterized these transcripts in that species itself. You'll have to rely on other species information. But that's the general technique, is to use other organisms to do your functional analysis.
Okay, great. Well, thank you so much for that answer. That was terrific. And so back to you, Yunlong. Here's a question: is in addition to variants in the promoter regions, what other factors can cause allele-specific expression?
Thanks for the question. That's a very good question. And actually, there are several recent publications and summarize the effects that can cause allele-specific expression differences. And promoter variants is apparently one of the most important ones and notable ones. Of course, this can be extended to all the other regulatory regions, including the enhancer regions, or in the 3' UTR regions, where the microRNA binding will occur there and to do the regulation for the gene expression levels. So meaning that if there is a variance in the 3' UTR region and the two alleles, they are different, and microRNA may be more functional in one allele compared to the other one, and therefore causing the allele-specific expression to happen. And but these are the variants in the regulatory regions in general. And there's another potential effect of allele-specific expression, which will be allele-specific epigenetic patterns, which is the including the DNA methylation, histone modification, and the chromatin accessibility. And there's a recent science publication and talking about the roles of allele-specific chromatin structure on the allele-specific expression, and those are the studies that need to be paid attention to.
Okay, great, excellent. So I was just going to ask anyone if there are any more questions. That would be terrific, but I'll ask another one to call in right now. So are there any specific issues in performing alignment with de novo transcriptome assemblies?
Great, thanks. So yeah, so in general with RNA-Seq alignment, if you have a reference genome, then you have a couple of choices. One is to actually align your reads directly to the genome, in which case you have to use these what are sort of referred to as splice-aware aligners that know that there exist these reads that might span a splice junction. Or you can do what we often try to do, which is just actually construct the transcript sequences itself and then align the reads straight to the transcript sequences. And therefore, you don't have to worry about these splice junctions. So in the de novo assembly case, that's what we tend to do, is to just align directly to the transcript sequences. And in that case, you don't need to have a very special aligner, just one that just tries to align the short reads to a reference using only maybe mismatches and indels. But one thing that does come up very often with these de novo transcriptome assemblies is that some of these assemblers are not very good at what we would call scaffolding, which is to sort of connect different assembled contigs together using paired end data. And then if that's the case, then you have to be very careful about actually not using the paired end data to do your quantification or transcript alignment, because many of the paired end reads will not align successfully because only one read will align to a contig. The other read might align somewhere else, and that would not be considered a successful alignment. So right now, depending on the assembler, we're sort of urging people to only use maybe one end of their data to do the quantification until we can get these assemblers all working nicely with the paired end data.
Okay, excellent. So I'm going to ask you both the same question, and I'll let you go first, Colin. It's just sort of, where do you see your research going over the next while, or or another way to look at that is what major questions do you think need to be addressed in the area in which you're working?
Sure. So one question that's really driving some work in my group right now is it has to do with these splicing graphs and also transcriptome assembly, which is when genes have many possible isoforms, how do we represent those isoforms, and what are the dependencies between splice events in isoforms? So there's a sort of an open question right now as to whether a splice event early in the gene affects the splicing later in the gene. And if there really is a strong dependence, then it makes sense to represent each gene by a sort of small set of isoforms, which is very popular right now. But if there's a lot of independence, then it actually means that we need to represent these things more abstractly as these graphs. And so that's something that's really driving my lab right now, is to try to tease apart exactly how much dependence is there between these splice events and how to represent that properly.
Okay, thank you. And over to you for the last question, Yunlong.
Yes, I think for my research interest, and as you mentioned earlier, it is construct computational models to really bring different levels of high throughput sequencing or functional genomics together, and to understand the process of gene regulation. That is the ultimate goal for my laboratory. And so far that we have trying to, so the talk today is to bring the DNA sequencing with RNA sequencing together with the transcription factor prediction algorithms to study gene regulation. And we also have some attempt to look at epigenetic effects by bringing in disulfide sequencing data and together with the DNA sequencing data, and to explain the RNA sequencing measured gene expression levels. So this type of integration is one of the focus of my research.
Great. Well, thank you so much to both of you today. We really appreciate it. And just want to remind everyone, we put up the survey link again if you could help us out and fill out that survey and let us know what topics you'd like covered in the future. And again, a big thank you to both Dr. Colin Dewey and Dr. Yunlong Liu for their presentations today. And we will be sending this out as a recording afterwards so everyone can listen to it and have an opportunity to learn more. So again, thank you all, and we appreciate your time today. Take care.
Thank you for the time.